Okay, thank you all for staying. And I'm actually not just going to talk about the host environment, oops, that was one too fast, and the ontogeny of the immune system, but focus specifically on the impact of host and environment interactions on the ontogeny, the development of the innate immune system. And I will do this mostly because of functional reasons, but also because I don't have time to talk about the whole immune system and its development. But most importantly, I am one of those specimens that Brett mentioned earlier on that comes to the study of the microbiome from a completely different angle, namely from reverse to what you've heard much about today. Namely, I'm a pediatric infectious disease physician. I take care of kids who die from infections. This is what it looks like. If on the x-axis you see age, roughly. On the y-axis you see incidence or cause of morbidity, mortality due to infections over life. And very obvious that infections as the cause of death and dying are the biggest problem the earlier in life. Then again, they creep up somewhere towards the end of life, and the shape kind of varies by global region, but the overall structure is the same. And if you look at the same kind of age distribution on the x-axis and look at the dogma as to what we believe is true for vaccine response, the dogma has it for most vaccines at least, that vaccines given early in life don't induce an immune response as effectively as the same vaccine given a little bit later in life. Somewhere around early childhood do you reach adult level of vaccine responses. That may not be true for all vaccines, but for the purpose of today's talk, that's sufficient. Unfortunately, most of the vaccines are given in early life, and that clearly is a discrepancy that is difficult to reconcile at the moment. But if you put these two observations together, increased incidence of infection, decreased vaccine response early in life, the interpretation has been, and that's the word that's used in almost all the papers talking about the development of the immune system, that the neonatal immune system or immune system in early life in general is immature. Immature means not ripe, not perfect. And for today's talk, I'm going to keep that and accept that word, although I strongly disagree with the word and the meaning of that word in this context. The reason that I focus on innate immunology today in this talk is that the innate immune system, to the largest degree at least, comes before the adaptive immune system. It instructs, as you've heard in several of the talks today, adaptive immunity, and then together they determine protection versus disease in the case of an infection. The function of innate immunity has to be also taken and looked at in the context of the genetic background of the host as well as the environment of the host. But broadly speaking, and you've heard much about this already, so I can skip through this, the innate immune system first and foremost functions as a sensor or a sentinel of the environment. It also produces effector molecules, for example, nitric oxide, as we've heard in the last talk. And then also, as I just mentioned, it regulates and interacts with the adaptive immune system in a bidirectional or multi-directional path. But basically, those are the three functions of the innate immune system. And so our hypothesis to begin with, knowing that early life has increased infectious incidence and decreased vaccine response, could it be that these differences are due to differences in innate sensor function, because that's the first step in this whole cascade between early and adult life. And just a quick notion, you've heard this already, that obviously if you imagine that this blue thing in the middle is an antigen-presenting cell, dendritic cell, only after stimulation through either pathogens or some pathogen molecules like PSA, as you just heard, are the functions, for example, in this case, antigen presentation, co-stimulation, cytokine production in this cartoon induced. The major function of these receptors that bind either specific molecules or whole pathogen, and therefore they're called pattern recognition receptors, are to identify these what has been called most often pathogen-associated molecular patterns. And I think you've already heard in several of the talks today that they're not necessarily pathogen-associated, they may be just microbial-associated patterns or even danger-associated patterns. But in any case, the function of these pattern recognition receptors is to activate or to inactivate, depending on which receptor is bound, the immune system, but to sense the immune system, to sense the environment. So the innate immune system really is a sentinel. Being a sentinel, sensitively sensing the environment, you're also very much prone to artifacts. And it took us several years to set up a system in which we can study the innate immune system with stabilized artifacts, let's say it that way. So we've really come up with a nice stable system that now is in use in multiple cohorts around the globe and has been performing pretty stably. And if you're interested in the details, I'd be happy to explain them. But everything that goes into the system is pre-tested, and you buy a whole lot that's enough for the whole study, and you complete the study and have very stringent SOPs and QC/QA procedures in place. So our hypothesis to start out with was that the innate response to, in this case, we focused on toll-like receptors, differs between early and adult life. And we approached this using cell-specific multichromatic or multiplexed flow cytometry, looking at the most important antigen-presenting cells, and also global cytokine screens using Luminix or ELISA systems. And for the purpose of the data that I'm showing you today, we did this in a cohort of about 100 children, all of them born in Vancouver, all of them born by cesarean section without sign of any inflammation around birth. We also completed a genetic screen on them, SNP screens of a total of about 750 SNPs, all positioned within the innate pathways that we were interested, including some ancestry verification SNPs. We collected the epidemiological data, growth, infections, vaccines, and all that like. And we phenotyped the innate as well as the adaptive immune system. And we collected stool samples at multiple time points. So these are distal intestinal samples, fecal samples. Arguably, as you heard, most of the immune reaction may actually happen in the small proximal bowel, but we can't sample that so readily. And we included the 2-year time point of the fecal sample, namely because, and you've heard this today already, that fecal composition, microbiota composition, early in life changes rapidly and fluctuates quite a bit, while it slowly stabilizes around 18 months. And so we figured picking a 2-year sample should give us a pretty much steady state screen. So we did all of this, and I'm going to show you none of the details of the immune phenotype. I'm going to focus on the microbiome phenotype. So the summary of the immune phenotype is in this slide, and caution, as it says on the slide, is this is not drawn to scale and hopelessly oversimplified. But if you imagine that, or if you look at the x-axis, we looked at preterm babies from about 23 to 24 gestational weeks of birth until full term, and then followed them for 2 years of age, until 2 years of age in the same cohort, and compared this to adult. If you look at this on the y-axis, you have the hypothetical level of production, for example, for IL-10, which is really the only cytokine you can induce in blood innate cells with any of the TLR stimuli that we've used before birth, meaning in babies that were born prematurely. That's cord blood at this point. And only once the baby is born at term. At term, can you induce huge amounts of IL-6 and IL-12P40 using many, not all, of the toll-like receptor stimuli. And the IL-12P40 parallels the production of IL-23. Interestingly enough, all of them, IL-6 and IL-12P40 and IL-23, drop off over the first 2 years of life, different kinetics, but they all drop down to almost non-detectable levels. IL-23 stays down, but IL-6 and IL-12P40 rise again sometime between 2 years and adulthood. While other cytokines, for example, TNF-alpha and IL-1 beta, generic pro-inflammatory cytokines, slowly increase over the first few months of life to reach adulthood levels by about 1 year of age, while type 1 interferon is produced almost at adult levels in the same response setup within almost 2 weeks to 4 weeks after birth. Contrary, IL-12P70, which slowly increases over the first 2 years of life, sometime between 2 years of life and adulthood, only then reaches adulthood, and interferon gamma, even at 2 years of age, has barely moved towards an adulthood level production. So this looks like a little bit of a spaghetti ball. If you put this together with the functions that we know that these various cytokines have if they're produced by innate immune cells, overall IL-10 production early on before birth is largely known, as you've heard again today as well, to drive overall immune suppression or at least an anti-inflammatory response, while the cytokines IL-6, IL-12P40 in combination with IL-12P19 make IL-23 are really driving what is known as the Th17 to fight extracellular microbes, and the peak production of these innate cytokines supporting Th17 development is right at birth, while antiviral cytokines rapidly mature within the first month, roughly. Pro-inflammatory cytokines in general take some time, and cytokines that are necessary to fight other intracellular microbes take a lot longer to mature. Now, putting this even on a more simplified scale and a more risky proposition, really, if you imagine this is now your immune development, and this is age from prenatal to term birth to early childhood, and you put a few specific numbers, namely birth 1 month and about 2 to 5 year toddler age right there, and then you correlate this, you graph with this, what are the major infections that we struggle with most of the time? Namely, E. coli, which is a regular extracellular bacterium, is a major pathogen in all babies that are born prematurely. Not the only one, certainly, but a major one. And it somehow stops for babies born at term. It doesn't become such a big player anymore. While HSV, that's herpes simplex virus, and listeria are both intracellular pathogens that plague neonates until about a month to 6 weeks of age, just about the time when your type 1 interferon defense reaches the maximum that I just showed you before, and afterwards they disappear as major pathogens. And on the other hand, tuberculosis or Burkholderia pseudomallei are both intracellular pathogens that the defense against which is highly dependent on production of IL-12P70, more specifically interferon gamma. And they actually plague you until somewhere between 2 and 5 years of age, depending on which cohort you look at. So the patterns of generic pathogen susceptibility correlates almost to the dot with what we find as a generic pattern of innate cytokine production over the first few weeks to months to years of life. And we call these periods windows of vulnerability. Now, with this, we've answered our initial hypothesis. There is a difference in innate cytokine production to TLR stimulation between early and adult life. That's pretty simple. And this is just a summary of what I've just shown you, that ontogeny, meaning the development of the toll-like receptor response, is regulated for specific toll-like receptors in specific cytokines in a cell-specific manner and in an apparently clinical relevant pattern. And this suggests underlying complex regulatory mechanisms that satisfy age-specific functional demands. And this is why I think the word immature, which means not ripe, not perfect, is inappropriate to use in this context, because it satisfies the neonatal or early infant demands. Out of this comes the clear hypothesis that age-specific changes are due to regulatory mechanisms that are aimed to optimize survival early in life. And this hypothesis really asks two specific questions, namely the regulatory mechanisms and survival early on in life, which is basically asking how does this happen and why does it happen? The how is relatively easy, and we're deeply involved in dissecting the molecular mechanisms that are involved in this. The why is a little bit more difficult to understand, and that's what I'm going to focus on today. So if you imagine, again, this is term birth, and this on the x-axis is age, and development of immunity on the y-axis in arbitrary units from prenatal to term to early toddlerhood. The Y for prenatal developmental suppression, namely IL-10 production, makes perfect sense because, as you all are aware, mom, baby are semi-allogeneic. They should be rejecting each other, and most often so far, at least as we talk about term babies, they're not. So having an early intrauterine existence where your innate immune system is geared towards anti-inflammation makes perfect sense. So the why there, at least, there's some reason to believe that we understand that. But we really have absolutely no clue what drives postnatal development of the innate immune system. Functionally, we have no clue. So the question then comes, what regulates the postnatal regulatory mechanisms? And again, looking at this very simplified generic diagram of the immune system functioning in the background of the host genetics and the environment, we were looking at the environment in a very generic term, and in all the cohorts that we're actively involved in, we're collecting multiple different angles or aspects of the environment. One of them is the gut microbiome. And with the gut microbiome, and you've heard this today already, you get a little bit more complex because this interaction between environment and host, or the innate immune system in this case, may be actually bidirectional, not unidirectional. I'll get back to this in a minute. So this is then what leads me to the topic for today, which is the host-environment interactions and how they impact the ontogeny of the innate immune system. The way we did this with the stool microbiome, just a few words on the technicalities, and if I say anything wrong, please ask Bill Moen to give you the correct answer, because I will fail. So we looked at the stool, and I'm only going to show you the stool for the 2-year age sample, because I told you before the fluctuations are tremendous, although they are interesting, and focus only on the 2-year stable, presumably, microbiome. And we did this using a phylochip that's commercially available, and the reason we use that is it's commercially available. That doesn't mean it's the best, and we're together with Bill and a few others, we're actually comparing different aspects. So basically it's an expression array or an array-based hybridization where you have your genomic DNA, you amplify the 16S RNA gene, you fragment, you label, and then you hybridize, and signal intensity gives you the readout. For the phylochip that we used from Second Genome, that's the name of the company, they have a total of 58,000 OTUs on that chip, and that's an important number to keep in mind for a minute. So what they then do is they take this, just as Bill outlined through the pipeline that they've established, seems to be working in their hands. They have multiple different pre-processing and data reduction steps. Then they measure either abundance, which is basically signal intensity, how often is a certain type of microbe present, versus binary, meaning that this sample is not present in a sample or it is present in a sample, independent of its abundance. Then you see how variable and how much overlap there is, and then you apply different components, and this is the one that that I'm going to show you today. It's basically similar to your principal component analysis. Of note, out of the 58,000 total OTUs that are present on the chip, only 13,000 were detectable in all of the 2-year samples that we looked at. And with this now, the first question that we asked in this simplified unidirectional interaction, is there correlation between innate immunity at birth and the composition of the gut microbiome 2 years later? So is the host determining the development of the gut microbiome? And this is just different TLR stimuli, different cytokines, the binary meaning yes, no, in those two different strain is present or not, versus abundance metrics. And to our surprise, even though we only looked at 30 subjects, we found 2 that were, at least according to the statistics that we calculated here, statistically significantly different. We weren't even looking for statistics. This was just too much of a surprise. If you look at that graphically, so if you cluster all the microbiomes according to family compositions, and that's where Bill would better be able to tell you what that actually means, with how much gamma interferon they made in response to Toll-7/8 stimulation at birth. So these are the microbial families present in the gut at 2 years of age versus how much interferon gamma or IL-10 they make in response to Toll-7/8 stimulation at birth as one of the ligands. You can see that they actually cluster. And strangely enough, the interferon gamma and the IL-10 cluster together, which they don't with the other cytokines. So not only do families of microbes in their composition seem to correlate with innate immune cytokine responsiveness in cord blood at birth, it seems that the cytokine production at birth in its composition, namely gamma interferon and IL-10, co-correlate with the structure of the family of the microbiota at 2 years of age. So the answer to our question whether there is a correlation between innate immune status at birth and gut microbiome at 2 years of age is yes. Although it has to be specified that this is true only for some cytokines after some stimuli. That's not true for every cytokine after every stimuli, and certainly not for all the OTUs that we had on the chip. If that's true, then the question, and this was brought up multiple times today, of chicken and egg, which comes first? Is it that the innate immunity drives the development of the gut microbiome, or is the gut microbiome driving the development of the innate immunity? And that's a question that we're struggling with, and I'm hoping that you will help me find out where we should go from here. So the question really then is who influences whom, when, where, and how? And again, getting back to this simplified graph, trying to come up with the chicken and egg question, we thought ourselves, well, this should be pretty simple, but environment isn't a simple term, just like the microbiome isn't a simple term to divide it into different categories, and neither is genetics. And what I mean with this is, in the simplified version, you could argue that you could classify environment and host or genetics into, for example, somebody of Chinese descent growing up in China or in Canada, and a Canadian growing up in China or in Canada. And it's a simple 2x2, and this way you have host and environment simply matched. But as it's apparent to everyone, I think, is the word Chinese doesn't tell you somebody is of this genetic makeup. Neither is a Canadian a simple Canadian. There's multiple different ethnic backgrounds that make this mix. So it's a lot more complicated than we had thought. And to address this, what we've started doing is set up cohorts similar to the one that I just described to you, where the data that I'm showing you today, in multiple parts of the world. So not just in Canada, but the US as well, Brazil, Ecuador specifically. We've completed the study in South Africa, and we're in the process of completing it in Belgium. That's barely visible right there. We're looking at multiple other parts, including Saudi Arabia and Thailand, and now also China, and there's a few others that will join, where we're going to look at exactly the same pattern that I just described to you in the Canadian kids in other parts of the world. Each of these cohorts have their own little sub-question, but they're all going to have the same generic summary of an immune ontogeny. So the first part then that we try to focus on in this complex interaction is ethnicity. And the question we asked is, if you compare cytokine production at birth between different ethnic groups, is there a difference? And again, focusing just on Toll-7/8 stimulation for gamma interferon, IL-10, TNF-alpha, and IL-12 in Caucasians or Asians. And that's just as a simple matter right now, just to show that there are some cytokines after this stimulation where there's no difference. There's some where the Caucasians make more, there's some where the Asians make more. And there's a mix of different patterns that emerge. If you then look at this is all at birth. If you then look at the gut microbiome of the same kids that we looked at for this innate response at birth and followed them over time, and look at them what they look like on a binary scale versus the abundance scale, you can see very clearly that the Caucasians and the Southeast Asians, that's the SA, the purple dot right there, all cluster in this space, or cloud as Bill called them, while the Asians all cluster around this cloud, and the Asian Caucasians mixed down here. This is true if you include 149 taxa out of the 5,800 that are, out of the 5,800 that we detected, and include when there is a sample present in more than 40%. And if you do this by intensity, even though this doesn't look like this, it still is statistically significant. So it's both binary as well as abundance-based, statistically significant that they cluster. Doesn't mean that this is the answer, but it means that at least within this case, for example, 150, just roughly, taxa of the stool of a 2-year-old child would allow you to predict that they would fall into a certain ethnicity. So the answer to the first question, do taxa distribution at 2 years of age correlate with specific subject ethnic origin, is yes. But again, as before, this is true for some of the distal gut microbiome, for some of the cytokines. And to move this up a little bit more towards a harder way to assay this, as I mentioned to you before, we included about 750 SNPs. Most of them are actually tag SNPs or positional SNPs, and looked at the impact of these SNPs on the innate immune ontogeny, again, looking only at birth right now and stool at 2 years of age, even though we have the time points in between. And we found the following pattern for just one of the SNPs that was interesting, which is the interferon response factor 7, or ERF7. That's the specific SNP number, and it's actually a missense coding SNP. And at birth, 1 and 2 years of age in the same group of kids followed interferon alpha production after Toll-7/8 stimulation. And you can see that those kids that have a phenotype of AA and those kids that have a phenotype of AG, both with respect to Toll-7/8 or Toll-9 stimulation, diverge by age 1. And in fact, they do so already by age 2 weeks and 4 weeks after birth. So that this difference in this, for this specific SNP, clearly identifies a specific genetic position, or genetic region at least, that influences type 1 interferon production at birth. And does that same SNP correlate with specific microbiome composition at 2 years of age? And the answer is yes. It's the same SNP, AA, AG, and you can see how the AA cluster over here and the AG cluster over here. That's true for binary, and this includes only 39 taxa. It's also true based on the statistics for the abundance measurement, including 267 taxa. So with this, the answer, do taxa distributions correlate, or microbiome composition correlate with specific genotypes and innate immune function? The answer is yes. But again, that's true for some of the microbial compositions, not for all of them, and only for some of the cytokines that we measured. So if you sum this all up, so ontogeny of the TLR response shows us a clinical relevant pattern. So we're looking at something that probably is important. Don't have proof for that, but it looks important. There's a correlation between innate immunity in birth and microbial composition, distal gut at 2 years of age. There is a correlation with ethnicity, and there's a correlation with specific SNPs, specific genetic regions. All of this then really puts this together for us, the chicken and egg. It probably argues that this is clearly a bidirectional interaction, but it's very difficult at the moment. This seems to be a stronger axis where genetics influences innate immunity, because that was present at birth before the baby was colonized. There already were differences in innate immune reactivity at birth, and that this drives the composition of the environment. And to answer this clearly, we have to look at the development of the microbiome over time and analyze the samples in between, which is in progress right now. And it's clear from what you've heard in the other talks today that there's multiple other ways in which the host microbiome can be influenced, not just through the innate immune system, clearly also through the adaptive immune system, and as well as through direct genetic effects on multiple other angles that we've not included in that. As I mentioned in the beginning, what we're looking at, and that's the data I'm showing you, is only 30 out of the 100 kids that we followed in Vancouver. And we have, if you add them all up, about another close to 1,000 kids spread around the rest of the world. So I've only shown you 30 out of the 100 right there. And we're using this as our training test to develop the hypotheses that A, make the most sense, B, are the most worthwhile to look at in detail, and then really use the rest of the sample set that we have to test specific hypotheses. And given everything that I've shown you today, I would be very glad if you can help us figure out which direction, which angles would be most fruitful in your opinion. And with that, I'd like to first of all thank both Brett and Philippe for inviting me to this really fantastic colloquium here already. And I'd like to also thank the NIH for funding, and the specific people who've done the microbiome work is Dr. Fortuno and Nelly Amogonu in the lab. And then Kevin, he's not on this, who's done, oh, there he is, on the analysis, while Nathan has done most of the work on the innate ontogeny, and with it also be open to questions if there are any. Thank you.